Hi folks, today I'm going to be talking about genetic disorders, specifically how do we categorize them and what makes them different from one another. We're not going to be able to spend a whole bunch of time talking about every single genetic disorder out there because there's far too many out there. But by categorizing them, we can see some similarities in the way that they're inherited from one person to another and exactly how are they caused in the cell, in the biological makeup of the genetic material. Specifically, a genetic disorder is one that is caused by abnormalities in some part of the genetic material. We know that our genetic material is DNA, so we could have a piece of DNA that is mutated just by changing one single nucleotide in the sequence, or we could have all the DNA bundled up together into a chromosome and have a piece of that chromosome be misplaced or missing or something like that. There's also genetic material in the mitochondria of our cells, and that's another way we can get that passed down. And we can also develop genetic disorders in something called epigenetics, which you'll get to study way, way further in the future there, where something about our environment changes our genetic makeup and causes a mutation, which is why we call it epigenetics, because epi means outside, so therefore it is from outside changing genetic material. Kind of cool there. So one category that we're dealing with is called a single gene disorder. A single gene disorder is caused by changes and mutations that occur in the DNA sequence of one specific gene. For example, GFP is a gene. Picture a paragraph of information, long paragraph that has a bunch of sentences together. Well, each one of those words in the sentence would be like a gene in a genetic sequence. And we know that if I change one word in a paragraph, the whole thing doesn't really read correctly. Well, that's the same way with a gene. If I change a gene or change something about a gene and try to read the genetic material to make a protein, well, that protein is going to be made with incorrect instructions and therefore have a different shape and different function because of it. A good example of a single gene disorder would be something like sickle cell anemia, where one specific mutation in a single gene causes a round erythrocyte to suddenly go from round shape to more of a sickle shaped, and therefore not be able to carry as much oxygen, and also tend to increase clotting factors and stuff. Really bad disease there. Now, single gene disorders are going to be inherited in a couple of recognizable patterns. You've heard these terms before, but I want to talk about them very briefly here. We know that autosomal dominant means that it's showing up in the normal chromosomes, not the sex chromosomes, the XX and XY. And by saying dominant, we know that it shows up quite frequently in the family. That's because that particular protein that is made by that genetic disorder is going to mask the correctly made protein, if there is one, and therefore the phenotype, that is the trait that we see in the person, will automatically show up. A good example of this would be hypercholesterolemia from PBS. You remember that you have an individual who is making too much cholesterol or having too much cholesterol saved and stored in their body. Well, that is automatically going to mask any other thing that happens in the body dealing with cholesterol, so it's autosomal dominant, and it will be passed on from generation to generation unless you all of a sudden have an individual who is a heterozygote and a homozygous recessive individual. Don't worry about that. We're not looking into inheritance. An autosomal recessive disorder, on the other hand, like albinism, is something that only shows up if the dominant allele is not there to mask the recessive one. So if an individual has a gene for albinism and has a gene to become albino, well, they can only really show albinism and show the albino skin if they don't have a gene for normal skin. Finally, there is sex-linked gene disorders, meaning that an individual would pass this down through the sex chromosomes and therefore a specific mutation on the sex chromosome's genetic sequence. And again, because it's sex-linked, it's going to be more common in specific ones, kind of like X-linked disorders are more common in males because we have the ability to only show one X chromosome, and therefore if the gene has a problem, it's going to show up in the next generation. I want you to contrast that with a multifactorial disorder, where it's not just a single gene that's been mutated, but instead we're looking at multiple factors that could cause this particular disease. Something along the lines of heart disease, for example. We know that individuals who smoke and don't exercise and are obese are more likely to have heart disease because of the environmental factors that associate with heart disease. But we also know that if you have a family history of heart disease, then you're more likely to have heart disease too. So there must be multiple genes associated with the inheritance of heart disease in addition to the environmental factors that we have in play. That means that most of our chronic illnesses are going to be multifactorial. Cancers, for example, are multifactorial. Heart disease, diabetes, all these different things that we know that a family history is related to the likelihood of us getting that disease, well, that's going to be something that is multifactorial. Now, chromosomal disorder is specifically related to the 46 chromosomes that we have inside of our body cells. Every single one of our cells, except for our gametes, the sex cells, have 46 chromosomes. So because these chromosomes carry a tremendous amount of genetic information, problems can quickly arise when there's an extra chromosome or an extra part of a chromosome or missing chromosomes or a significant break in a chromosome or even just a switching of a part of a chromosome. So we in PBS looked at karyotypes to be able to diagnose chromosomal disorders, because all we had to do was look at the structure of each chromosome, try to find its matching pair, and figure out, is one section missing or is there an extra one in play. Finally, a mitochondrial disorder is relatively rare, but it is something that can happen with mutations found in the mitochondria. Your mitochondria has its own set of DNA inside of it. It's non-chromosomal because it's not bundled up into the 26 shapes or anything like that. And it's unique because this mitochondrial DNA that's locked inside there is only going to be passed from the mother to the child. That's because the mitochondria from the sperm cells are basically destroyed by the time that the sperm cell gets into the egg, whereas the egg, its mitochondria, last pretty long through the gestation process, through the development process of a zygote. That's it for our 4 types of genetic disorders. You're going to have one of these 4 types for your disease, for your patient. So I advise you to get used to being able to say these particular things so you can give your patient, through telehealth practice, more of an idea of how this is passed down through generations.